Hello, I'm Professor Sims, and in this video, I will discuss the cellular level of organization. This is the first in a series of 10 lessons held as part of my Anatomy and Physiology 1 course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. In this unit, we will learn about the structure and function of the cell membrane, the functions of various cytoplasmic organelles, the structure of nucleus, the process of DNA replication, the process by which a cell builds proteins using the DNA code, stages of the cell cycle including cell division and somatic cells, how a cell differentiates and becomes more specialized, and the morphological and physiological characteristics of some representative cell types in the human body. All living cells and multicellular organisms share a common feature, the cell membrane, which acts as a protective barrier regulating material passage. Structurally, the cell membrane is composed of a flexible bilayer of phospholipids interspersed with cholesterol and various proteins. The phospholipid molecules have hydrophilic phosphate heads and hydrophobic lipid tails, creating an amphipathic structure. The membrane's fluidity allows proteins embedded in it to perform functions like facilitating transport. Integral proteins embedded in the membrane include channels for selective substance passage and cell recognition proteins. Peripheral proteins found on the surface carry out specific cellular functions. Figure 3.2 illustrates the structure of phospholipids that make up a cellular membrane. The phosphate head is polar and hydrophilic, which means water-loving. The tail is made of saturated and unsaturated fatty acids that are nonpolar and hydrophobic, which means water-hating. Figure 3.3 shows the phospholipid bilayer. Notice that the two layers of phospholipids align tail to tail, and their hydrophilic heads form the exterior of the membrane. This is where they can interact with fluids both inside and outside of the cell. Figure 3.4 shows the cell membrane with the phospholipid bilayer as well as several other types of molecular components, such as glycoproteins, glycolipids, cholesterol, and integral membrane proteins, peripheral membrane proteins, and channel proteins.
All cells are completely surrounded by a membrane that separates them from their environment, kind of like how your skin completely covers your body. This cell membrane is sometimes called the plasma membrane, and it acts as a barrier, regulating what gets in and out of the cell. Because the membrane lets some things through but not others, we say that the membrane is semi-permeable or selectively permeable. If we zoom in on the membrane, we can see that it is made of several molecular parts. Most of the membrane is made of molecules called phospholipids. Phospholipids have a head made of phosphate and two tails made of fatty acids. The heads are hydrophilic, meaning that they are attracted to water, or literally, water-loving. The tails, however, are hydrophobic, meaning that they repel water. Literally, hydrophobic means water-fearing. Because of these properties, phospholipids form two layers known as a phospholipid bilayer, with the hydrophobic tails facing each other and the hydrophilic heads facing the watery external environment and the watery cytoplasm inside of the cell. So the water lovers get to be next to water, and the water fearers can hide away from it. A perfect arrangement. The phospholipid bilayer is the main component of the membrane, and it is largely responsible for making the membrane semi-permeable. Basically, the smaller and less charged something is, the easier it is to pass through the phospholipid bilayer. Bigger, more charged things have a harder time. A second membrane component is cholesterol. Although people often think of cholesterol as a bad thing, it's actually a very important part of cell membranes, where it helps to stabilize the membrane and make it better at controlling what can pass through it. Proteins are another important part of the membrane. They do many functions, but one of the most important ones is transporting materials across the membrane, especially if they have a hard time passing through the phospholipid bilayer. A fourth membrane component is carbohydrates. These are often involved in cell identification. Blood types, for example, are the result of membrane carbohydrates attached to proteins. These help you to identify blood that belongs to you and distinguish it from foreign invaders. All of these components work together to make the plasma membrane that surrounds all cells, a critically important barrier needed for all life on Earth.
The cell membrane's selective permeability permits the passage of small, nonpolar substances through simple diffusion. while larger polar molecules require facilitated diffusion. Water, through osmosis, moves freely across the membrane. Active transport, requiring ATP, moves substances against their concentration gradients, often involving pumps like the sodium-potassium pump. Secondary active transport utilizes energy created by primary pumps for the movement of other substances. Additionally, endocytosis allows the cells to ingest material by enveloping it in membrane portions, forming vesicles. Phagocytosis and pinocytosis bring in larger amounts of extracellular material, while receptor-mediated endocytosis is more selective. Conversely, exocytosis involves vesicle transport for the secretion of cellular products into the interstitial fluid. Figure 3.5 shows how small uncharged molecules can move through the semipermeable cell membrane via simple diffusion. This type of movement through the membrane doesn't require the assistance of integral membrane proteins, as the solutes simply migrate down their concentration gradient. Facilitated diffusion, as shown in Figure 3.6, is another form of passive diffusion that requires the assistance of channel proteins and carrier proteins. This passive diffusion does not require an input of energy, and molecules are moving along their concentration gradient. There's two types of channel proteins. You have open channel proteins and gated channel proteins. Open channel proteins create a pore in the cell membrane and allow the charged molecules to pass through. The gated channel proteins close or open to regulate movement across the membrane based on size and charge. Carrier proteins carry the molecules, change their conformation, and then release them to the other side of the membrane. Carrier proteins are more selective, often only allowing one particular type of molecule to cross the membrane. The sodium-potassium pump is a membrane protein responsible for maintaining the electrochemical gradient across the cell membrane. It actively transports sodium ions out of the cell and potassium ions into the cell against their respective concentration gradients, utilizing energy derived from the hydrolysis of adenosine triphosphate, or ATP. The net result is the active transport of 3 sodium ions out of the cell and 2 potassium ions into the cell during each cycle. This creates an electrochemical gradient because there are more positively charged ions inside the cell and more negatively charged ions outside of the cell. And this gradient is important for various cellular processes, including maintaining the resting membrane potential, electrical excitability of nerve and muscle cells, and the regulation of cell volume. Figure 3.10 illustrates the 3 forms of endocytosis. Each form of endocytosis contributes to the cell's ability to regulate its internal environment, acquire nutrients, and remove waste. Phagocytosis, or cellular eating, is primarily performed by specialized cells such as macrophages and neutrophils. The cell membrane extends projections called pseudopodia around the particle. These pseudopodia fuse, forming a vesicle called a phagosome, which encloses the ingested material. The phagosome then fuses with lysosomes containing digestive enzymes, forming a phagolysosome. Digestive enzymes break down the engulfed material, and the residual waste is expelled from the cell. Pinocytosis, or cellular drinking, involves the nonspecific uptake of small dissolved molecules and extracellular fluid. The cell membrane invaginates, forming small vesicles called pinocytic vesicles. These vesicles enclose the fluid and solutes, bringing them into the cell. In receptor-mediated endocytosis, ligands in the extracellular fluid bind to receptor proteins on the cell's surface. This binding triggers the formation of coated pits, which are regions of the membrane coated with a protein called clathrin. The coated pit invaginates, forming a coated vesicle that contains the ligands bound to receptors. The vesicle then detaches from the membrane and fuses with endosomes. The ligands are released from their receptors, and the receptors are recycled back to the cell surface or sent to lysosomes for degradation. Receptor-mediated endocytosis is highly efficient and allows cells to selectively internalize specific substances such as hormones, cholesterol, and nutrients. Figures 311 and 312 show the process of exocytosis and the formation of secretory vesicles. Exocytosis is how cells expel or secrete materials from their interior to the external environment. This mechanism involves the fusion of vesicles, which are membrane-bound sacs with the cell membrane, to discharge their contents. This process is important for a range of physiological processes. In neurons, for example, neurotransmitters are released from synaptic vesicles into the synaptic cleft, allowing communication between nerve cells. In endocrine cells, hormones are secreted into the bloodstream through exocytosis. Cells use exocytosis to renew and modify their membrane composition and to eliminate waste materials. The process is tightly regulated and plays a fundamental role in maintaining cellular homeostasis and communication with the external environment. Now let's explore the internal components of cells and their roles. Within the cytoplasmic compartment of all multicellular organisms, we'll find jelly-like substance called cytosol. Cytosol provides the necessary fluid medium for biochemical reactions. Eukaryotic cells house various cellular organelles, each serving a specific function and collectively contributing to the overall health and functionality of the cell. The endomembrane system, comprising the endoplasmic reticulum, or ER, Golgi apparatus, and vesicles, executes the production, packaging, and export of specific cellular products. The ER facilitates material transport, synthesis, and storage. The rough ER is studded with ribosomes and focuses on protein synthesis, while the smooth ER specializes in lipid synthesis, detoxification, and carbohydrate metabolism. Following protein synthesis in the rough ER, the Golgi apparatus, which resembles stacked discs, sorts, modifies, and dispatches products, acting kind of like a cellular post office. Lysosomes contain digestive enzymes that break down cellular components and foreign materials. They also engage in autolysis and controlled cell death, known as apoptosis. Mitochondria, known as the energy transformers or the powerhouse of the cell, convert nutrients into ATP, providing cellular energy. Peroxisomes, akin to miniature sewage treatment plants, handle lipid metabolism, detoxification, and neutralization of harmful reactive oxygen that cause oxidative stress and disease in certain species. Figure 313 shows an animal cell with all of its organelles. So this would be a eukaryotic cell. We know that because it has membrane-bound organelles. We have vacuoles, so the cytoplasm, Golgi apparatus. Then we have the nucleus, the nucleolus, chromatin, rough ER with the ribosomes, smooth ER on the outside of that doesn't have ribosomes, lysosomes, peroxisomes, mitochondria, and of course the plasma membrane. Figures 3.14 and 3.15 provide illustrations and electron micrographs of both the smooth and rough endoplasmic reticulum. Note that the main structural difference is the presence or absence of ribosomes, which are the sites of protein synthesis. Figures 3.16 and 3.17 show the structures of mitochondria and peroxisomes. Mitochondria produce energy, while peroxisomes detoxify the cell and metabolize lipids. The cytoskeleton, comparable to the bony skeleton supporting humans, provides structural integrity to cells. The cytoskeleton helps to maintain cell structure, supports motility, aids in cell reproduction, and facilitates substance transport within the cell. Figure 3.18 shows the three components of the cytoskeleton: microfilaments, intermediate microfilaments, and microtubules. Microfilaments are composed of long, thin fibers of actin protein. They're about 7 nanometers in diameter and are used in cell motility, cytokinesis, and maintaining cell shape. Microfilaments are responsible for cell crawling and muscle contraction, and they can undergo rapid assembly and disassembly. Intermediate filaments are coiled proteins such as keratin, vimentin, and lamin. They're about 10 nanometers in diameter and provide mechanical strength to cells and tissues. Intermediate filaments are less dynamic than microfilaments and microtubules, having limited involvement in cellular movement. Microtubules are hollow tubes made from tubulin protein. They're about 25 nanometers in diameter and form a rigid internal skeleton that acts as tracks for intercellular transport. Microtubules are essential for ciliary and flagellar movement, and they form the mitotic spindle during cell cell division.
The cytoskeleton, as the name implies, is the skeleton of the cell. The cytoskeleton of a cell is a network of filaments running throughout the cell. These filaments have different jobs in the cell. They help provide support, much like your skeleton. They also provide tracks or highways for the movements of vesicles and other objects. They help pull the chromosomes apart during mitosis, and sometimes even help the cells move. Ribosomes are tiny protein factories. They receive instructions from the DNA and converts this information into a chain of amino acids, which becomes a protein. Each type of protein has a different chain of amino acids. Next, we have the nucleus, and it's the location of the genetic material and DNA of a cell. It sends out instructions to other organelles, much like the ribosomes. The rough endoplasmic reticulum, or rough ER, is involved in protein production and transporting proteins. It is the location of many ribosomes, which makes it look rough. It takes these proteins created by the ribosomes, and it sometimes may modify these proteins and then packages them into a vesicle and sends it out to the Golgi apparatus. Smooth ER, which is smooth because it does not have ribosomes, produces lipids, which are fats, plus helps to detoxify the cell. The mitochondria, it's the powerhouse of the cell. It creates this energy by taking glucose plus oxygen and makes the energy molecule ATP. Vacuoles are organelles that store materials like water and other materials. You gotta have storage. Lysosomes, which are created at the Golgi apparatus, they contain enzymes that help break down waste materials and digest materials in the cell like bacteria. Speaking of the Golgi apparatus modifies and sends out proteins and lipids into the cell. It receives these proteins, modifies them, tags them, and then sends them either into the cytoplasm or maybe to the cell membrane where they are released out of the cell.
The nucleus is the largest organelle in a cell, and it serves as the central hub for genetic instructions governing protein synthesis. While certain cells, like muscles, may be multinucleated, others, like red blood cells, don't have any nucleus at all. Enclosed by a nuclear envelope with pores, the nucleus contains a gel-like nucleoplasm housing a nucleolus responsible for ribosomal RNA synthesis. Chromatin threads within the nucleus carry genetic information in the form of DNA, which condenses into chromosomes during cell division. Figure 3.19 shows the location of the nucleolus, chromatin, and nuclear envelope that make up the nucleus of animal cells. And figure 3.20 gives an example of cells that contain more than one nucleus. These are multinucleated skeletal muscle cells, also known as muscle fibers. And they differ from cardiac muscle cells because cardiac muscle cells only have one nucleus. Figure 3.21 shows red blood cells that do not have a nucleus. Red blood cells undergo a maturation process in the bone marrow, during which they lose their nucleus and other organelles. This process, called enucleation, results in cells that are specialized for oxygen transport via hemoglobin, and this is an important adaptation for a specific physiological role in the circulatory system. DNA consists of two antiparallel strands of alternating sugar-phosphate backbones with complementary base pairs forming the interior. Adenine pairs with thymine and cytosine pairs with guanine, and that's complementary bases. DNA replication is a process required for cell reproduction, and it encompasses initiation, elongation, and termination stages, ensuring the conservation of half of the original DNA in each new molecule. DNA proofreading mechanisms correct errors, maintaining genetic integrity. This meticulous replication process is vital for preserving the accurate transmission of genetic material during cell division. Figure 3.23 shows the structure of DNA. So it is an antiparallel strand of alternating sugar-phosphate backbones with complementary base pairs on the inside, right? The nitrogenous base pairs inside are going to be either thymine, guanine, adenine, or cytosine. And each one of these nitrogenous bases has a sugar attached, a deoxyribose sugar, and a phosphate group. And these are what make up the backbone on the outside. Figure 3.24 gives an overview of the process of DNA replication in animal cells. During DNA replication, the double-stranded DNA molecule unwinds with the help of enzymes, and it creates two template strands. DNA polymerase synthesizes complementary strands extending from the 3' to 5' direction and produces two identical DNA molecules. The process is semi-conservative, with each resulting DNA double helix consisting of one original strand and one newly synthesized strand.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
DNA acts as the cell's blueprint, holding vital information for protein construction. Genes, functional DNA segments, contain the code for protein synthesis. A cell's proteome is its complete set of proteins, and it stems from the genes. The interpretation of genes involves the translation of DNA bases into an amino acid sequence, and then the amino acid sequence encodes the specific amino acids required for protein synthesis. The process begins with transcription, creating a complementary mRNA strand, mRNA being messenger RNA. The mRNA undergoes modification, including splicing, before leaving the nucleus. Translation follows, with mRNA serving as a template for protein synthesis in the ribosomes. The tRNA ferry the corresponding amino acids to the ribosome. The tRNA or transfer RNA aligns them according to the mRNA codons, and the process concludes when the mRNA's final codon signals termination, releasing the newly synthesized protein. Figure 3.25 gives a schematic overview of transcription and translation. In short, the nucleotide sequence, that is the sequence of adenosines, cytosines, guanines, and thymines in DNA, is transcribed into RNA, which is then translated into specific amino acids that make up a protein. Figure 3.26 provides another overview of transcription from DNA to messenger RNA via RNA transcript and the enzyme RNA polymerase. This is the first stage of making a protein from DNA. The second stage is translation, where the messenger RNA is read by the ribosome, and transfer RNA molecules. Transfer RNAs match specific amino acid sequences to the mRNA sequences. Figure 3.29 illustrates how transcription occurs within the cell nucleus, while translation occurs outside of the nucleus in the cytoplasm.
Now that we understand the structure of DNA, it's time to understand exactly how this molecule codes for a particular organism. How is it that a single cell containing a specific set of genetic material will result in the development of a fish, or a cat, or a human? To understand this phenomenon, we have to learn about transcription and translation. This is the collective process by which the genetic code is read by enzymes in order to produce all of the proteins in an organism. A chromosome is a very long molecule, consisting of many millions of base pairs. Most of these bases don't do too much, but certain portions of the chromosome are special. They are called genes. These are the parts that code for different things. In a human, a gene will be on average around 10 to 50,000 base pairs long, though the longest is 2.5 million base pairs. And when a gene is expressed, a specific protein is produced. So how does this work? The first step is called transcription. This is the process by which enzymes use one of the strands of DNA within a gene as a template to produce a messenger RNA, or mRNA. To do this, RNA polymerase, with the help of proteins called transcription factors, binds to a specific sequence within the gene, which is called the promoter, and pries the two strands apart. One of the strands will serve as the template strand, or antisense strand, meaning it will be used to generate the mRNA, and the other is the non-template strand, or the sense strand. RNA polymerase doesn't need a primer. It simply initiates mRNA synthesis at the start point and then moves downstream along the gene in a process called elongation, synthesizing the mRNA as it goes, reading the antisense strand from 3' to 5' and generating the mRNA from the 5' end, attaching RNA nucleotides to the 3' end as it goes. This is very similar to the way DNA polymerase synthesizes DNA as it moves along the template strand. The main difference here is that RNA is being synthesized, which, as we recall, will be ribose rather than deoxyribose, and it will have uracil instead of thymine. Unlike replication, RNA polymerase zips DNA back up as it goes, keeping only 10 to 20 bases exposed at a time. Once RNA polymerase reaches the end of the gene, termination occurs. The enzyme detaches from the gene and the DNA is returned to its original state. But we have produced an mRNA. This carries with it the information encoded in the gene, and after a few quick modifications during RNA processing, it will leave the nucleus, where all the genetic material, or chromatin, is, and move into the cytoplasm, where it will find a ribosome. This is where translation occurs. During translation, the mRNA acts as a code for a specific protein. This happens because each set of 3 bases on the mRNA, which we call codons, will code for a specific anticodon, which will be carried by a specific transfer RNA, or tRNA. And each different tRNA is covalently linked to a particular amino acid. The arrangement of the nucleotides into these codons is called the reading frame. Since there are 4 bases and each codon has 3 letters, 4 cubed gives us 64 different possible codons, which is more than enough to code for all the amino acids we need. Here is a table of all the mRNA codons and the amino acids they code for. Notice that there is some redundancy, with multiple codons resulting in the same amino acid, but there is no ambiguity. Each codon corresponds to a particular amino acid. Notice also that some of these codons are special. AUG is the start codon, which initiates translation by coding for methionine, and these 3 are stop codons. These are the ones that terminate translation. Translation will occur inside a ribosome. The small ribosomal subunit binds to an mRNA and an initiator tRNA, which adheres to the start codon. Then the large ribosomal subunit joins to complete the translation initiation complex. Then the tRNA that corresponds to the next codon after the start codon will enter the ribosome. This will carry with it an amino acid, which becomes covalently bound to the methionine from the initiator tRNA. The first tRNA detaches and leaves the ribosome, which has shifted over, making room for the next tRNA. The new amino acid links to the first 2, and this process continues all the way down the mRNA. As tRNAs enter and exit the ribosome in a sequence that is dictated by the codons on the mRNA, a polypeptide chain will grow. This continues until a stop codon is reached, at which point the completed polypeptide will swim away, most likely entering one of the cell organelles for folding and further modification. So in this 2-step process, DNA is transcribed into an mRNA, and then this mRNA is translated into a protein, all simply by obeying the base pairing that occurs in nucleic acids. And since every gene codes for a specific protein, and proteins make up most of what you are, from your muscle tissue and organ tissue to all of your receptors and enzymes, this is how DNA carries the code for a living organism.
The cell cycle orchestrates cell life from creation to division and comprises interphase, mitosis, and cytokinesis. Interphase involves distinct phases including G1 for growth, S for DNA synthesis, and G2 for preparing for mitosis, and G0 represents a resting phase. During the synthesis phase, DNA replicates, resulting in sister chromatids attached by a centromere. Mitosis encompasses prophase, metaphase, and anaphase and telophase, followed by cytokinesis, leading to the formation of two daughter cells. The cell cycle is tightly regulated by control mechanisms involving checkpoints and molecules such as cyclins and cyclin-dependent kinases. Checkpoints, including G1, G2, and metaphase, determine whether the cell should proceed or stop at specific phases. Cyclins and CDKs collaborate to push the cell cycle forward, ensuring that each phase's processes are completed before advancing to the next phase. Aberration in cell cycle control can result in cancer, where abnormal cells undergo uncontrolled division. Genetic predisposition and environmental factors often contribute to the failure of cell cycle controls. Figure 3.30 gives an overview of mitosis and interphase. Mitosis is where cell division occurs, while interphase is where the cells grow and perform all of their normal functions, like DNA synthesis. Figure 3.32 shows the stages of cell division via mitosis. The four main stages are prophase, metaphase, anaphase, and telophase. In prophase, chromatin condenses into visible chromosomes, the nuclear envelope disintegrates, and the mitotic spindle forms. During metaphase, chromosomes align along the metaphase plate. Anaphase involves the separation of sister chromatids pulled to opposite poles of the cell by spindle fibers. And telophase concludes mitosis with the formation of two new daughter nuclei, the uncoiling of chromosomes, and initiation of cytokinesis. Figure 3.33 gives an overview of how cyclins and cyclin-dependent kinases regulate cell division by forming complexes that control progression through the cell cycle. Cyclins are proteins whose levels rise and fall, and their binding to CDKs activates these kinases. The activated CDK cyclin complex triggers specific events such as DNA replication or mitosis by phosphorylating target proteins and thus regulating the cell cycle.
Cell growth and division are divided into phases. Cells grow during the G1 phase and the genome replicates during the S phase. During the G2 phase, cells continue to grow and prepare for cell division. During the M phase, mitosis occurs. Cytokinesis, C, is the phase when the cytoplasm divides, creating two daughter cells. Three principal checkpoints control the cell cycle in eukaryotes. The G1 checkpoint makes the key decision as to whether the cell should divide, delay division, or enter a resting stage. The G2 checkpoint assesses the success of DNA replication and triggers the start of the mitosis, M phase. If this checkpoint is passed, the cell initiates the beginning of mitosis. The accuracy of mitosis is assessed at the M checkpoint. This checkpoint occurs during metaphase and triggers the exit from mitosis and cytokinesis and the beginning of G1. At the G2 checkpoint, cyclin-dependent kinases, CDKs, phosphorylate histones and proteins that carry the cycle past the checkpoint into mitosis. During G2, the cell gradually accumulates G2 cyclin, also called mitotic cyclin. The cyclin binds to CDK to form a complex called MPF, mitosis promoting factor. When the level of MPF exceeds the threshold necessary to trigger mitosis, the G2 phase ends and mitosis begins. One of many functions of MPF is to activate proteins that destroy cyclin. As mitosis proceeds to the end of metaphase, CDK levels stay relatively constant, but G2 cyclin is degraded, causing progressively less MPF to be available and initiating the events that end mitosis. After mitosis, the gradual accumulation of new cyclins starts the next turn of the cell cycle. The G1 checkpoint is thought to be regulated in a similar fashion. The level of G1 cyclin increases and associates with cyclin-dependent kinase, CDK. Eventually, a threshold ratio that triggers the next round of DNA replication is reached. The cyclin is degraded and the cycle begins again.
The development of a complex organism from a single fertilized egg involves cellular differentiation, where unspecialized cells transform into specialized cells with distinct functions. Stem cells, characterized by their ability to divide and differentiate, are classified based on their potential. Totipotent stem cells have the potential to differentiate into any cell needed for an organism's growth. Pluripotent stem cells, derived from totipotent cells, can differentiate into any human tissue but cannot support full organism development. Multipotent stem cells are more specialized and can differentiate in specific cell types within a given lineage. Further specialization leads to oligopotent and unipotent cells. Stem cells found at different life stages include embryonic stem cells, fetal stem cells, and adult stem cells such as epithelial stem cells and bone marrow stem cells.
There are both internal and external cues for stem cells, which can involve these transcription factors. Examples? Okay, an example of an internal cue could be transcription factors present in the cytoplasm of the original starting zygote cell, which will eventually be present in the cells that come from it. Now, the specific location of the stem cell within the developing embryo can matter because the transcription factors available in different areas of the developing embryo can differ in quantity and type, which could impact what a stem cell differentiates into. External cues could involve cell signaling from other cells next to it. External cues can even be environmental effects like temperature. There's still a lot of research in this area, and we can't wait to see what scientists discover about this in the next decade. So, stem cells are the unspecialized, undifferentiated cells that can become other cells in your body. But not all stem cells are found in a developing embryo. Stem cells can also be found in your body as well, like your muscles, skin, liver, bone marrow, just to name a few. These are often called somatic stem cells. To give some relevance to this, it's likely you've heard of bone marrow transplants before. Well, bone marrow transplants actually involve transplanting a portion of healthy bone marrow, which does contain bone marrow stem cells, with the idea that those donor stem cells can help regenerate different types of blood cells, since bone marrow is like a blood cell-making machine. It contains stem cells that differentiate into different types of blood cells. Many, but not all, of the somatic stem cells that are found in your body are considered to be multipotent. That means they can become many types of cells, but not as many as the embryonic stem cells. So after talking about these stem cells, why the heavy focus on them right now in research? Well, one reason, of many, is that these cells have the ability to differentiate into other cells, and therefore they could be used to help regenerate organs or tissues that are damaged from a disease or an accident. There are two important issues to consider, however. One is the ethical issue, especially if considering embryonic stem cells. The ethical issue is significant because the extraction of embryonic stem cells results in the demise of the embryo. One point consistently debated is the potential benefits offered in embryonic stem cell research versus the onset of personhood of human embryos. A second issue is that organ or tissue developed from stem cells that didn't originate from that person will carry the risk of organ or tissue rejection, possibly similar to what you could get with donated organs or tissue. But here's something promising. Some research shows that somatic stem cells from your own body may actually be able to develop into more types of cells than what people first thought. In fact, it was discovered that some somatic stem cells can be induced to go back into a pluripotent state. They're called induced pluripotent stem cells. That means a person's own somatic stem cells from their own body could potentially be induced into a pluripotent state with the potential that they could differentiate into tissues or organs that the person may need. Theoretically, this could be an alternative to waiting for an organ or tissue donor, as well as lower the chances for organ tissue rejection, since the organ or tissue would have originated from the person's own cells.
Differentiation involves changes in size, shape, metabolic activity, and function, driven by the unique genetic expression of each cell. Transcription factors regulate gene activation or inhibition, directing cell differentiation. Stem cell research explores using these cells for cellular regeneration and repair. Adult stem cells existing in various tissues contribute to tissue renewal and repair. Inducing stem cells to differentiate in a laboratory setting offers potential treatment for several diseases. Figure 3.36 shows how stem cells have the unique ability to differentiate into various cell types, offering potential treatments for a range of diseases. Stem cells are often used in regenerative medicine, where they can be directed to replace or repair damaged tissues and organs. Stem cell therapies hold promise for conditions such as cardiovascular disorders, neurodegenerative diseases, and even certain types of cancers.
In the early embryo, very soon after fertilization, there are stem cells that are able to differentiate or mature into any other tissue in our body. These cells are therefore called pluripotent stem cells. But very quickly thereafter, they become specialized. They can only contribute to the tissue in which they live. These are tissue-specific stem cells. So stem cells in the brain only produce new brain cells, and those in the gut produce gut cells. Almost all our organs contain a small population of these tissue-specific stem cells. In every tissue, many cells die when they reach their normal lifespan or after injury. In some tissues, the lifespan of cells is very long. Cells in the heart, skeletal muscle, brain, and the lens in your eye all live very long. In contrast, in other tissues, the lifespan of cells is very short, such as skin, gut, and blood. In fact, every week, the inner lining of your gut is renewed, and every month your skin is replaced completely. This is possible by tissue-specific stem cells, which continuously replace cells that have become lost. One could argue that tissue stem cells are nature's own anti-aging agents. Without these stem cells, many tissues would age prematurely, as no repair could ever happen. However, whereas skin wounds recover very quickly in young infants, as we grow older, healing gets more difficult. Many old people suffer from too few blood cells and become susceptible to infections or anemic. It seems very plausible that at least some aspects of reduced tissue functioning during aging results from a reduced functioning of stem cells. How could stem cells age? As stem cells divide very often during the lifetime of an organism, they need to replicate their DNA and pass this on to their two daughter cells. Damaged DNA is therefore bad for stem cells. Patients that suffer from diseases that result in increased levels of DNA damage typically have problems with regenerating tissues and age prematurely. A special case of DNA damage relates to the ends of chromosomes, also referred to as telomeres. These ends shorten a little bit with every cell division. If the ends become too short, a stem cell cannot properly divide any longer. There may be multiple other reasons why stem cells perform worse with age. One of these appears to relate to the process of epigenetics. Epigenetics refers to the program that dictates which specific components of the DNA are used by a cell. For example, the hemoglobin gene is present in all cells of our body, but it is only used by red blood cells. Similarly, the insulin gene is only used by cells in the pancreas. DNA in a cell is typically very compact, and compact DNA cannot be used. If a cell needs to use a stretch of DNA, for example to produce hemoglobin or insulin, this part of the DNA needs to open up so that it becomes accessible. Epigenetic enzymes take care of this and locally open up or close stretches of DNA. Epigenic regulation can be compared with the instructions that a software program uses to select which components it needs at a specific time point to execute a certain function. It has become clear that these epigenic instructions are very important for stem cells, and for unclear reasons, change with age. This may result in some information becoming inaccessible in aged cells. At present, we do not know how to delay aging, but if we would be able to find ways to keep stem cells functioning properly during aging, tissues would remain more healthy.
Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics and leave your questions for me in the comments below.